All right, welcome back to another lecture on the nervous tissue unit. I think this is like number 4. Anyways, so last time we were going over the resting membrane potential. And also remember, one of these Edpuzzle lectures is going to be randomly selected to go under exam points. So what does that mean? So if you're kind of not watching this and the questions come up and you get half of them right, and I happen to pick this one for our exam, then you got 50% on a basically an open note, open Google, open everything exam. So you're going to want to watch it, take some notes, and get all the questions right just in case I pick this one for an exam. Anyways, so last time we were doing the resting membrane potential when the neuron is not firing an action potential. So that's right here. Remember, it was negative 70 millivolts. So that's this right here when the neuron's staying ready to fire an action potential. Remember, it did this through the work of the sodium-potassium pump. Remember, the sodium-potassium pump pumps out 3 positively charged sodiums and pumps in 2 positively charged potassium ions. Remember, you also had the leak channels. Remember, there's a lot more potassium leak channels that allow those potassium positively charged to leak out than there are sodium that it's allowed to leak in. So that also helps to keep it at negative 70 millivolts. Also, remember, inside the neuron, there's a bunch of negatively charged proteins, amino acids, and also inorganic phosphates. All right, so this lecture is not going to be on how the resting membrane potential is maintained, but this is where a neuron starts to get stimulated. So this is going to be what's called the action potential or the nerve impulse. So the neuron starts to get stimulated, and it's usually the dendrites. Remember, that's the receiving end. So it might be neurotransmitters from an adjacent or nearby neuron. The neurotransmitters are striking sensory receptors on the dendrites, and that's what's going to start depolarizing the neuron. And I'm going to show you how this works in a second. Depolarizing means it's less and less negative. So there's positive ions are moving into the neuron, and it's drifting away. It's drifting higher than that negative 70 resting membrane potential, the negative 70 millivolts. And it starts to drift higher and higher if it gets stimulated enough. If it reaches this right here, this is called the threshold. If it reaches this polarity, so if the polarity raises from negative 70 to negative 55, boom, it's an action potential, and that's it right here. So it goes all the way up to positive 30. Some neurons, they say positive 40. And then it's repolarized. Briefly, it's hyperpolarized, and then it returns back to the resting membrane potential. So the process of it going from negative 70 and becoming less negative, that's depolarization. If it reaches negative 55 millivolts, called the threshold, it's going to shoot. It's like a chain reaction. Once it hits negative 55, there's no stopping it. It's going to rush up inside the neuron all the way to positive 30, and then it's going to need to repolarize. It needs to get negative again in order to be ready to fire another action potential. So this is going to be the rush of positively charged ions out of the neuron in order to get it negatively charged again. And then briefly, it's actually charged below the negative 70. It could be negative 75, negative 80 millivolts. And that's called hyperpolarization. All right, so your first page of notes. Action potential, initiation, and generation. All right, so neurons respond to stimuli or a stimulus. So that's what excites a neuron and gets it more likely to fire an action potential, to fire a nerve impulse. Remember, the vast majority of your neurons are in your brain, and these are all interneurons. So they're interneurons talking with another interneuron across a synapse. And to transmit the signal across the synapse from presynaptic neuron to the postsynaptic neuron, it releases neurotransmitters. Those neurotransmitters cross the space, cross the synapse, and hit receptors on the postsynaptic neuron, and it starts to initiate an action potential or stimulate the postsynaptic neuron. But there are also sensory neurons. They're not stimulated by neurotransmitters. They have sensory receptors. So you have different receptors for light, sound waves, pressure in your cutaneous receptors, that's in your skin. You have receptors for itch. You have receptors for increased temperature, warmth, decreased temperature, cold. You have receptors for stretch, pain. So those are all different types of sensory receptors that will excite sensory neurons. But anyways, regardless of the stimulus, the result is always the same. Cell permeability changes for a short time in the neuron. All right, so what does it mean by the cell permeability changes? All right, so remember, you have the presynaptic neuron, axon terminal, synaptic end bulbs. They contain the neurotransmitters. Postsynaptic neuron, the dendrites. So the presynaptic neuron is going to release neurotransmitters into the synapse, and they're going to strike these little gates on the dendrites of the postsynaptic neuron. They're called ligand or ligand. I've heard it pronounced both ways. I like ligand better. Which one's totally correct? I don't know. Anyways, but when the neurotransmitter hits that ligand gated channel, it's going to open up and it's going to allow positively charged, usually positively charged sodium to start coming into the neuron. And since positively charged sodium is coming in, it's going to start moving off that resting membrane potential of -70 and start to become less negative, more towards the positive. So it's going to start getting depolarized. So that's what depolarization is. It's the influx of these positively charged ions, usually sodium. Some neurons use calcium ions, but usually it's sodium, and that's causing it to start becoming less negative, more going towards the positive region. And that's called depolarization. Now, the initial influx is called a graded potential. So this is not an action potential. This is where the neuron is starting to get stimulated, but it hasn't been stimulated enough in order to generate an action potential. So I can go back over here. So right here, resting membrane potential. All right, some neurotransmitters from a presynaptic neuron starts to strike the dendrites of the postsynaptic neuron. It starts allowing sodium in. This is a graded potential right here. It's not an action potential yet because it hasn't reached the -55 millivolts. So if that neuron doesn't get stimulated enough, it's going to go right back down into the resting membrane potential. But if it does get stimulated enough and it reaches the threshold of -55, now different type of channels open up. They're called voltage gated. And that's the ones that's going to cause or generate the action potential. All right, so if you can understand how these different channels or gates, how they work, that's over half the battle to understanding the action potential. All right, so first you have the ligand or ligand gated channels. These are stimulated by neurotransmitters. Neurotransmitters sent from the presynaptic neuron crossing the synapse, and it strikes the receptor on this ligand gated channel. So the neurotransmitter floats across the synapse, sets here in this ligand gated channel, and then it causes the channel to open up. And that's going to allow sodium to start diffusing into the neuron. And that's what causes this. This is what causes the depolarization of the neuron. So it's these ligand gated channels located on the dendrites. When the neurotransmitter binds with them, it opens up and allows sodium to start diffusing in to depolarize the neuron, making it move away from that -70 and becoming more towards the positive end. And down here you have the leak channels, and these aren't really important in generating an action potential. Remember, these are always open. These allow potassium to mainly diffuse out of the neuron from high concentration of potassium inside to low concentration of potassium outside. And fewer of these are sodium leak channels. And so those allow sodium to slowly leak into the neuron from high concentration of sodium outside to low concentration inside. So these are for helping to maintain the resting membrane potential, not for an action potential. All right, so over here we got mechanically gated channels. So these are not stimulated by chemical neurotransmitters. These are located at the ends, or the dendritic end, or the sensory end of your sensory neurons. So these are stimulated, open and closed, due to some sort of physical stimuli. So in this case, it's pressure, but it could be itch, it could be pain, it could be warmth, it could be cold, it could be stretch. A lot of different physical stimuli that can stimulate these mechanically gated channels. So in this instance, it's pressure. So it might be something pressing down on my skin. Well, why do I feel that? How can I feel that pressing down on my skin? Well, that's the pressure pressing on a sensory receptor in a sensory neuron that's causing my mechanically gated channels to open up. And that's what's causing or allowing positively charged, in this case, these are calcium ions, because these are what's called piezo channels that allow calcium to depolarize the neuron. So that's what's causing this to happen. Mechanically gated channels open up due to some sort of physical stimuli, and in this case, it allows calcium to come in. Now, you're not going to need to know piezo channels. The reason why I know you're not going to need to know it, I just learned it yesterday. I was reading about mechanically gated channels, and these are mostly depolarized by the inflow of calcium ions instead of sodium. But the vast majority of the time, it's going to be the sodium that causes this depolarization of the neuron. All right, and the last one is the real star of the show when it comes to the action potential, and that's the voltage gated ion channels. Voltage gated, so they're not stimulated by neurotransmitters. They're not stimulated by any kind of physical stimuli. They open and close when the membrane of the neuron reaches a specific voltage. So in this case, this is the sodium, voltage gated sodium channel, because I see Na plus right here. So when does it open? It's going to open right here at -55 millivolts. So remember, these guys, the mechanically gated, the ligand gated, They're stimulated by either neurotransmitters or some sort of physical stimuli. Those are the ones that take the neuron from negative 70 and start moving it towards what's called the threshold of negative 55. Now, if enough of these mechanically gated or ligand gated channels are open to allow enough sodium to diffuse in to reach that magic number of negative 55, boom. Now the voltage gated sodium channels swing open and sodium is allowed to rush into the neuron from high concentration outside the neuron to low concentration inside the neuron. And that's what causes this. That's what causes the polarity to go from negative 55, the threshold, all the way up to positive 30, because now the neuron is filled with positively charged sodium and filled with a positively charged potassium. So that's what an action potential is. It is reaching that threshold of negative 55 millivolts because enough of these mechanically gated or ligand gated channels allowed enough sodium or calcium to move in, reaching that magic number of negative 55 millivolts, causing the voltage gated sodium channels to open, which shoots up the polarity from the threshold of negative 55 all the way up to positive 30. Sometimes you'll see positive 40. It just depends on the neuron. All right, so we discussed how depolarization is caused by voltage-gated sodium channels being opened up at negative 55 millivolts, the threshold required. Well, there's not a picture for this, but how does the neuron actually repolarize? So how does it take it back down from positive 30 and get it back down to the resting membrane potential of negative 70 millivolts? Well, you can probably figure it out. So now inside the neuron, following this rushing in of sodium ions that cause this right here, now we have a bunch of sodium inside the neuron and we got a bunch of potassium. So how are we going to get it back negative? So we got to get some positively charged ions out of the neuron. So what happens when it starts reaching the peak? The voltage gated sodium channels are going to close. So now sodium can no longer move into the neuron. And when it reaches positive 30, voltage gated potassium channels are going to open. And now potassium, since there's a lot of potassium inside the neuron, very few of it outside the neuron, potassium is going to start rushing out of the neuron. Positively charged potassium rushing out of the neuron from high concentration of potassium inside to low concentration of potassium outside. And that flow of potassium ions outside the neuron is what brings the polarity or the voltage back down to negative 70 millivolts. All right, so a little review on the resting membrane potential. When a neuron is actively maintaining its negative 70 millivolts, getting ready to fire an action potential, but it's not actually firing one. So remember, outside the neuron is a positive charge, inside it's negatively charged, and it's negative 70 millivolts. Inside the neuron, the cytoplasm of the neuron, there's a bunch of potassium ions, high concentration of potassium. Remember, there's also proteins, amino acids, and organic phosphates, all which have a negative charge. That's what helps to contribute to that negative 70 millivolts inside the neuron. Extracellular, outside the neuron, high concentration of sodium compared to the inside. A lot of sodium on the outside, not much on the inside. Remember, there's leak channels that allow sodium to leak in, potassium to leak out, but there's way more potassium leak channels, so more of the positively charged potassium is able to leak out than positively charged sodium is able to leak in. And don't forget about the sodium-potassium pump, which actively, you remember, uses ATP energy to actively pump out 3 sodiums and pump in 2 potassiums, helping to maintain that resting membrane potential. All right, the action potential graph. So you see here the resting membrane potential, and here's a graded potential. Well, I mean, it looks like it reached negative 55, but I guess it didn't quite reach it. So remember, either a ligand gated or a mechanically gated channel is starting to get stimulated. It's allowing usually sodium to start diffusing into the neuron, and it's going to start depolarizing it, making it less negative. And here you can see that magic number, the threshold of negative 55 millivolts. If it gets stimulated enough to reach that, the voltage gated channels, sodium channels, are going to open and fire an action potential. All right, now some words for your notes. All right, depolarization. So this is a chemical or a mechanical stimuli that's causing the neuron to leave that resting membrane potential of negative 70 millivolts and start to creep less negative or slightly going towards the positive region. And this is due to allowing sodium ions to start diffusing into the neuron. So remember, neurotransmitters, it's the ligand gated channels. Neurotransmitter strikes it, it opens up, and sodium can start coming in, start to depolarize. Mechanically gated, some sort of physical stimuli that allows those to open up, allow either sodium or calcium to come in and start to depolarize the neuron. Remember, if it reaches the magic number of negative 55 millivolts, that's now going to cause the voltage-gated sodium channels to open. And that's what's going to cause this. The voltage gated channels that takes it from not from negative 70, but from negative 55, the threshold, up to positive 30 millivolts. And that's what the action potential is. All right, so this nerve impulse is going to start at the axon hillock. Remember, that's where an action potential is initiated. But this is what's called positive feedback. So once a segment or a region in the axon hillock reaches negative 55 millivolts and causes a sodium voltage gated sodium channel to open, it's positive feedback. It's like a chain reaction. So it's just like setting up a bunch of dominoes. You put dominoes all over, going up things. And then you push over that first domino. The first domino hits the second domino. And then it causes a chain reaction where all the dominoes end up falling over. Same thing with these voltage gated sodium channels. Once one of them opens up and it goes to a positive 30 inside the neuron, that's going to cause the next one to open up, which is going to cause the next one, the next one, the next one. And it goes continuously all the way down the axon. So that's how it transmits or propagates the nerve impulse all the way through the neuron, because one voltage gated sodium channel is going to cause the next one to open and then the next one, just like dominoes falling over. All right, so in that brief time where the neuron is firing an action potential, it is actually 1,000 times more permeable to sodium than when it is in its resting membrane potential. And it only lasts for about 1 millisecond, because that action potential is going to boop, boop. It's going to go, it's kind of like a heartbeat, boop, boop. But once it goes up, it needs to come right back down, and it's got to be ready to fire another action potential. So it's really, really fast. And remember that the membrane is going to shift from the resting membrane potential of negative 70. If it reaches the threshold of negative 55 millivolts, voltage gated sodium channels open, and that's going to shoot it past zero and all the way up to positive 30 millivolts. And now the outside is going to be more negative, and the inside is going to be more positive, because now the inside has it had a bunch of potassium already, but now a bunch of sodium came in. So now it's got a bunch of potassium and sodium. And that's what causes it to shoot all the way up to positive 30 millivolts. And it's that spike right there. That's what's called the action potential. All right, now the graph again. Remember, here's the resting membrane potential, negative 70 millivolts. I can actually draw a straighter line than this one. But anyways, so ligand-gated or mechanically gated channels start to get stimulated by neurotransmitters or some sort of physical stimuli, and it starts to depolarize. This is a graded potential. Hasn't reached negative 55 yet. If it gets stimulated enough, now remember, if it doesn't get stimulated enough, it's just going to go right back down to the resting membrane potential. But if it does get stimulated enough, and it reaches that magic number of negative 55 millivolts, the threshold, now the voltage gated sodium channels are going to spring open, and it's going to shoot up because of the sodium rushing in all the way up to positive 30 millivolts. All right, so in the action potential, resting membrane potential, the neuron starts to get stimulated. It reaches negative 55 millivolts, and it starts to shoot up due to the inflow of sodium ions into the neuron. But once it passes zero and starts to peak at 30, now it's going to become, the neuron's going to be, it's going to decrease in sodium permeability. That's caused by those voltage gated sodium channels. As that voltage starts to get up close to 30, passes zero and starts to get into the positive, they start to shut. So now sodium's like, hey, I can't get in anymore. So since sodium can no longer diffuse in, that's what causes it to peak. It doesn't go any higher than positive 30 millivolts. All right, so now the process of repolarization. So first we got the resting membrane potential, negative 70 millivolts. It starts to get depolarized. It reaches negative 55. Boom, we get an action potential, fully depolarized. But now we're way up here. We got to get that neuron back to its resting membrane potential of negative 70 millivolts. Well, how do we get it back down? How do we repolarize the neuron so it's ready to fire another action potential? Well, once it reaches the peak, that's where sodium entry is going to decline. So sodium coming in is going to be halted. But right here at the top of the mountain, that's where voltage gated potassium channels are going to open up. So they open up at around positive 30 millivolts. That's when those channels go open up. And that's going to cause potassium to rush out of the cell from high concentration of potassium inside the cell to low concentration of potassium outside the cell. And since potassium has a positive charge, it moving out of the neuron is going to bring it back down. It's going to repolarize the neuron. And it actually is going to be even below negative 70 for a brief moment. And that's called hyperpolarization, before the sodium potassium pumps kick in and get it back to that resting membrane potential of negative 70 millivolts. And here's the graph I just drew, but it's in the slide, so I'm going to show it. All right, once again, negative 70 millivolts, the resting membrane potential. Ligand-gated channels or mechanically gated channels start to allow sodium to diffuse in because of some sort of stimulation, either a neurotransmitter or some sort of physical stimulus. If it reaches a threshold of negative 55 millivolts, voltage-gated sodium channels will open up, allowing sodium to rush into the neuron. Now, once it starts getting past zero and starts becoming positive, voltage-gated sodium channels will close. Sodium can no longer come in, so it levels off. Voltage-gated potassium channels open, so potassium starts to rush out of the neuron, and that's what repolarizes the neuron and brings it back down to negative 70 millivolts. All right, now hyperpolarization. All right, so I'm just going to show it in a little graph I'm going to draw, so I won't show you the next graph, I don't think. Anyways, remember, once again, resting membrane potential. Maybe if you hear it like 15 times, it'll stick in the brain. Negative 70 millivolts. And then ligand-gated or mechanically gated channels start to allow sodium to creep in. If it reaches negative 55 millivolts, voltage-gated sodium channels will open and it'll shoot up to positive 30 millivolts. Sodium channels close. No more sodium can come in. Voltage-gated potassium channels open. Potassium starts to rush out of the neuron. They're positively charged, so that's what brings it back down. But as you can see, potassium permeability continues. So those gates are sluggish. They're slow to shut. So it reaches negative 70, but it can't like just go and slam on the brakes. It's slow. So it actually goes a little under negative 70, maybe negative 75, maybe negative 80. And then the sodium potassium pumps kick in and brings it right back to the resting membrane potential of negative 70 millivolts. So right here, that's hyperpolarization. Now, I don't know if this will be on the test, but when the neuron is hyperpolarized, that's what's called it's called a refractory period, and it's specifically the relative refractory period. You might want to write that down. So when the neuron is hyperpolarized, right in this section right here, it's a relative refractory period. The neuron can fire an action potential, but it's going to need a very strong stimulus. So it may not feel like a little tight little tap. It's going to need a kind of a strong stimulus in order for it to fire an action potential. Now, there's another refractory period, and it's called the absolute refractory period. I don't know, it's not in our book, but it should be. Anyways, the absolute refractory period is right here. So it's right in this region right here. So it's when the graph is going down. So it's when the potassium channels are open and potassium rushing out of the neuron, and this graph is going down before it hits the hyperpolarization and starts going back up. In the absolute refractory period, the neuron cannot fire an action potential no matter how strong the stimulus. So the relative refractory period, the neuron can fire another action potential, a second action potential, but it has to be a strong stimulus, a lot of neurotransmitters, for example. But in the absolute refractory period, when this graph is going down, no matter how strong the stimulus, the neuron cannot fire an action potential. It's not ready yet. All right, some more words for your notes. All right, so what we got here? All right, so repolarization, it restores the resting electrical conditions. So we had depolarization, sodium moving in, repolarization, potassium moving out of the neuron. So the potassium moving out did restore the negative 70 millivolts, the electrical conditions, but it did not restore the original ion distribution, because now we got a bunch of sodium inside the neuron when it's supposed to be outside. And we got a bunch of potassium outside the neuron when it's supposed to be inside. That's where Mr. Sodium Potassium Pump kicks in. Remember, so sodium potassium pump's going to start pumping out 3. There's a bunch of these guys. They require a lot of energy. They start pumping out 3 sodium. So sodium, get out of here. You need to get out of the neuron. Potassium, you need to get back in. So it's the sodium potassium pump that restores the ion distribution. So repolarization, that's due to potassium moving out. That restores the electrical conditions. But it's the sodium potassium pump that restores the ion distribution inside and outside the neuron, making more sodium outside and more potassium on the inside. All right, I want to show this diagram of the process of what's called summation at the axon hillock. So remember, the axon hillock is this tapered region. It's like the first part of the axon or the last part of the cell body in this box right here. All right, so you can see these are what's called excitatory neurons, these green ones. These are the axon terminals and synaptic end bulbs of the presynaptic neuron. And here's your postsynaptic neuron. So these guys, so the synapses are right here. This is the dendrites. So these guys are releasing what's called excitatory neurotransmitters. And it's called the excitatory postsynaptic potential. So that's in green here. So when these guys release neurotransmitters, it starts to depolarize the neuron. It allows sodium to move in, and it starts to creep closer to that negative 55 millivolts. But you also see these pink or purple ones right here. These are inhibitory neurons. Inhibit. So they're not excitatory, they're inhibitory postsynaptic potential. Now, when these guys release their neurotransmitters, it's usually a neurotransmitter called GABA, GABA. It's an inhibitory. It brings a neuron further away from an action potential. It actually polarizes it even further. So it brings it down. It doesn't depolarize going up. It actually makes it repolarizes going down. So you can see it happen again. So this neuron right here, it's getting stimulated by a excitatory presynaptic neuron, excitatory postsynaptic potential. And that's what's going to start depolarizing the neuron. But then right here, another inhibitory neuron starts to release its neurotransmitters. And it actually causes the neuron to become more negative, repolarize. And it does this by two ways. So inhibitory neurons inhibit a neuron by two ways. So either one, and this is the direct route. Let me make this a little smaller. Make it a little bigger than that. In the direct route, so when these neurotransmitters strike the neuron, the ligand-gated channels of the postsynaptic neuron, it's going to allow Cl minus ions to enter the cell. So that's going to cause, when Cl minus ions enter the cell, since they're negatively charged, that's what causes it to repolarize or become more negative. But another way it can do it is the indirect route. This is where same neurotransmitter GABA, but it has to go through a different step. And it allows, when it strikes the ligand-gated channel, it's going to send a secondary messenger to another channel, and allows K plus ions to move out of the neuron. So since positively charged potassiums are leaving the neuron, this also helps to inhibit. Doesn't allow the neuron to fire an action potential. So either one of these, the inflow of chloride ions or the outflow of potassium ions, those are both inhibitory postsynaptic potentials because it doesn't depolarize the neuron, making it go up. It repolarizes, or it can even hyperpolarize the neuron by making it go down. So this all occurs, summation all occurs at the axon hillock. So this neuron is adding up all the excitatory postsynaptic potentials that it's receiving from the excitatory neurons, and it's also adding up all the inhibitory stimulation from these inhibitory presynaptic neurons. And if when they add it all up at the axon hillock, if it reaches negative 55 millivolts, boom, you'll get an action potential. If it does not reach it, you will not have an action potential. The neuron will simply go right back down to its resting membrane potential. So this is summation. It's the adding up of excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Excitatory stimulation, inhibitory stimulation. You add it up. If it equals negative 55 millivolts or higher, you're going to get an action potential. If it doesn't reach that magic number, no action potential will be fired. All right, I don't know if this is supposed to be the last slide, but I'm going to make it the last slide because we're getting on close to 30 minutes. But once again, what do they call it in college? Just getting started, just getting warmed up. All right, so neurons work on what's called the all or none response. What that means is, let's see. All right, so a neuron, there's no such thing as a strong action potential or a weak action potential. All action potentials are the same. So there's not like hey, that's a weak action potential for like maybe a slight, like a little touch. And then if somebody really hits you with a hammer or something, then you have a strong action potential. No. All action potentials are the same. So it's not like it goes up to negative or positive 100 millivolts for a strong stimulus. All are the same. They either fire an action potential or they do not fire one. So now the question is, how does your body differentiate? Tell the difference between a light touch and someone just hammering you and maybe breaking a bone. How do your neurons, since there's no strong action potentials, how do you differentiate between something that doesn't really hurt versus something that really, really hurts? Well, it's not the strength or the amplitude of the action potential, but it's the frequency. So something that you can feel it, but it doesn't really hurt. It's not a very strong stimulus. It'll be something like this: resting potential, action potential, and then a little while later, action potential, and a little while later, action potential. So that tells your brain, okay, I feel it, but it's not a very strong stimulus. Now, if I put gasoline on my arm and light it on fire, that's a very strong stimulus. That's not going to be action potential. That's going to be like this, action potential. It's going to be a very high frequency of action potentials, kind of like if your heart's going really fast, like dee, 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 dee. So that's how your neurons tell your brain the different type of stimulus. So if it's a very light, not a very strong stimulus, it'll be action potential. Or if it's really strong stimulus, it's going to be very, very fast frequency of action potentials. And your brain responds to that as like, whoa, either that really hurts or it's really high pressure, but it knows that it's a very, very strong stimulus. All right, I hope some of this made sense, and I'll see you next time for lecture number 5.